Hi everybody. In this unit we're going to continue learning about propagation from seed. After learning about the factors affecting seed germination and steps we can take to optimize germination in the last unit, we're now going to look more closely at seed that doesn't germinate readily, even when we give it appropriate levels of light, water, and temperature. By the end of this unit, you'll be able to describe methods for overcoming dormancy in seeds, and you'll know what some of the best resources are for looking up protocols for overcoming dormancy. And you'll use hot water and mechanical scarification techniques on seeds with physical dormancy in a lab exercise. So let's get started. This first slide is just a list of some of the resources I use most frequently when I want to look up the treatment protocols for breaking dormancy in seed, or I want to put together some research to come up with my own protocols. I put this slide at the beginning of this lecture because invariably I'm asked halfway through the lecture, where can we find all the protocols for overcoming dormancy in seeds? You can find a copy of this slide in Canvas in the Supplemental Resources section for this module. There are, of course, many other sources of information, and there's also no substitute for networking and staying up to date with what's going on in the industry by going to conferences and watching webinars. My own favorite organization and annual conference for learning about propagation and plant production is the International Plant Propagators Society. Let's start with a couple of definitions. We learned in the previous unit that a non-dormant seed is one that has the capacity to germinate readily when it's given physical environmental conditions of temperature, light, water, and oxygen that are normal for that particular species or genotype. And this type of seed is described as being in a state of quiescence. In contrast, a dormant seed can be defined as one that doesn't have the capacity to germinate in a specified period of time under any combination of normal physical environmental factors that include temperature, light, water, and oxygen that are otherwise favorable for its germination. So why does seed dormancy exist? I mentioned in the previous unit that most commercial seed has been bred and sometimes pre-treated so that it germinates readily when we provide the appropriate environmental conditions. Seed dormancy can be frustrating or challenging for a grower, but in the wild, it's one of nature's survival mechanisms. Dormancy prevents seed from germinating when the environmental conditions aren't conducive to successful seedling establishment. Imagine yourself as a plant that grows in subalpine areas of the Sierra Nevada, for example. Your seed ripens and is dispersed at the end of summer. There may be sufficient early rain in the fall for your seed to germinate and emerge, but is your seedling going to survive the freezing temperatures of winter and probably being buried under several feet of snow? No. So it doesn't behoove your seed to germinate as soon as there's sufficient water. It benefits your seed to develop a state of dormancy and not germinate until the spring, when the soil will still be moist, but temperatures will be warmer and your seedling stands a much better chance of survival as it establishes itself over the summer months. Remember I mentioned in the introductory unit to this class that a knowledge of plant ecology can be important in propagation. This is particularly applicable when we're working with wild collected seed or seed that we haven't worked with before, when it may be important to know what plant community the plant we're wanting to grow is from, and what the climate's like in the area from which the seed was collected. This knowledge can help us determine what treatments we're going to give our seed in order to overcome any dormancy. As with many of the propagation techniques that we talk about over the course of the semester, it's important to remember that there's no one treatment we can use for every plant, and that treatments for overcoming dormancy may vary according to the age of the seed, weather conditions when the seed was forming, and they may differ between different ecotypes of the same species. Despite advances in plant science, we still know very little about the physiological processes involved in seed germination, seed dormancy, and overcoming seed dormancy. We do know that dormancy is controlled by the physiological or structural properties of a seed, and that it also responds to external environmental conditions. The natural plant growth regulators, abscisic acid and gibberellic acid, appear to be important in inducing and stimulating germination, but the amounts of these two biochemicals and plants' responses to them aren't consistent across all species. And the techniques we use to break seed dormancy are based more on empirical evidence rather than scientific knowledge at the moment. And as I mentioned earlier, no single technique works with all dormant seed, and in fact the same technique may not even work consistently from year to year with the same genotype. Let's look at the different classes of seed dormancy. We've known about the mechanisms of seed dormancy for many years, but it wasn't until 2004 when Dr. Jerry Baskin and Dr. Carol Baskin from the University of Kentucky, and who are two of the world's top plant scientists whose research is focused on seed, proposed a formal classification system for seed dormancy that has been adopted worldwide by plant scientists. The Baskins proposed a system with 5 major classes of seed dormancy: physical, morphological, physiological, morphophysiological, and combinational dormancy. And within each of these classes, they proposed further subdivisions of levels and types. Seed doesn't always fit neatly into just one of these classes and can exhibit more than one class of dormancy. We're going to take a quick look now at each of these 5 classes, and then look at some of the techniques we can use in the nursery and at home to overcome seed dormancy. Let's look at physical dormancy first. This is probably the simplest of the dormancy classes, both in terms of understanding it and overcoming it. As seed ripens, the seed coat hardens and becomes water impermeable and/or gas impermeable. In the wild, this is a survival mechanism that prevents all seeds from germinating at once next season and ensures that some seeds remain in the soil seed bank as a backup. Examples of seeds with physical dormancy include many species in the pea family, the Fabaceae, so lupins, redbuds, and acacia. Other examples include bladderpod, Peritoma arborea, which you'll be working with in the lab exercise. The photo on the right is of the California native shrub yellow bush lupin, which is one of the other seeds that you'll be working with in the lab exercise. In seeds with morphological dormancy, the embryo is underdeveloped at the time that the seed is disseminated. The seed isn't physiologically dormant, it just needs extra time to finish maturing and will usually germinate normally, given the right environmental conditions, within 30 days after imbibition, the first stage of water uptake in the germination process. Examples of seed that have morphological dormancy are our California native, Western azalea, and kiwi fruit, and blue honeysuckle or haskap, which is a shrub that produces fruit that looks like elongated blueberries and has been gaining in popularity in the US in recent years, in line with the increasing interest in edible landscaping. Physiological dormancy is characterized by the presence of a fully developed embryo, unlike morphological dormancy, and it usually takes more than 30 days after imbibition for the radicle to emerge. In their classification system, the Baskins identified varying levels or depths of physiological dormancy. One type of physiological dormancy is epicotyl dormancy, where the radicle emerges without any treatment, but the epicotyl, the shoot, doesn't emerge until it's undergone a period of chilling. If we go back to the subalpine plants in the Sierra Nevada example that I talked about at the beginning of this unit, then this might be a typical example of epicotyl dormancy. Imagine yourself again as that plant in the subalpine Sierra Nevada. It certainly isn't advantageous for your shoot to emerge just before the onset of winter, but it may well be beneficial for your root to get established during the fall and winter months so that it can quickly supply your shoot when it does emerge in spring with water and nutrients. Seeds exhibiting physiological dormancy may just need several months of cold stratification, or they may need a combination of treatments, including gibberellic acid, stratification, and scarification. The photo on the right is of Calicarpa americana, American beauty bush, a shrub identified by the Baskins as having physiological dormancy. This is a shrub that you rarely see in California landscapes, although you occasionally see cultivars of its Asian relative, Calicarpa bodnieri, which is also grown for its showy clusters of pinkish purple berries in the fall. And I think Suncrest Nursery in Watsonville may be growing a couple of these cultivars. In morphophysiological dormancy, we have a combination of the previous two classes of dormancy that I talked about, the morphological and physiological dormancy. This is a combination of the two, where the embryo isn't fully developed at the time of seed dissemination, and there's an inhibitory physiological factor. Overcoming dormancy in these seeds usually involves providing more than one treatment in succession. This slide just shows pictures of what the plant whose seed was pictured in the previous slide looks like. This is Ambarella trichopoda. It's a plant that you'll probably not see in any California ornamental landscape, but it's a plant which is particularly fascinating at the moment because it's being used in a lot of research that's being done on the evolution of plants, and in particular the evolutionary history of seed dormancy in the angiosperms. Let's look now at combination dormancy. Combination dormancy is the term applied to seeds that have both physical dormancy and the embryo is physiologically dormant. In these seeds, after the impermeable seed coat is ruptured, the seeds still need further treatment to overcome physiological dormancy. An example of this is the plant that's pictured here on the right. This is one of our locally native Ceanothus species, and you can see this in the wild if you go hiking up at Quail Hollow County Park. This is Ceanothus papillosus, variety Roweanus, the wart-leaf Ceanothus. And seed of this Ceanothus needs scarification to remove the physical dormancy, followed by between 75 and 90 days of cold stratification in the fridge. Secondary dormancy isn't one of the classes proposed in the Baskin's classification system. Secondary dormancy is a term applied when seed fails to germinate after primary dormancy is broken, because the seed is exposed to unfavorable environmental conditions. In nature, this is an additional survival mechanism. For example, imagine yourself as a plant that's native to a desert ecosystem. Your seed has developed and is lying quiescent in the soil. There are a couple of days of rain and your seed starts to germinate. It begins the imbibition process, the initial uptake of water that's going to activate enzymes in the seed to continue the germination process. But suddenly the rains stop and there's a heat wave when ambient temperatures are in excess of 100 degrees Fahrenheit. The chances of the seedling from this seed surviving if germination and emergence continue are probably zero. So therefore, the imbibed seed goes into a state of secondary dormancy, which can sometimes be really difficult to overcome. Usually it requires a period of chilling to break secondary dormancy, and there may also be other treatments that need to be given to break secondary dormancy. A common example of a plant that exhibits secondary dormancy is lettuce, which is sensitive to high temperatures in excess of the range of 26 to 35 degrees Celsius. So this is well above 80 degrees Fahrenheit. And lettuce seed will go into a secondary state of heat-induced dormancy. Growers can avoid this by purchasing seed that's been primed, and that was one of the additional services that's often provided by commercial seed growers that was mentioned in the previous unit. Let's look now at some of the techniques we can use in a nursery or at home for breaking seed dormancy. Firstly, let's look at scarification. Scarify means just to roughen the surface of something. And scarification techniques are used to break physical dormancy in seeds that have a hard, water impermeable and or gas impermeable seed coat or a waxy seed coat. Techniques that we can use include mechanical abrasion, hot water soak, fire, and acid. And these techniques mimic what would happen in the wild. For example, wildfire, a seed passing through the acidic gut of a berry or seed-eating bird or other animal, or the effect of soil microbial activity breaking down the seed coat. Examples of seed that usually need scarification are lupins, bladderpod, redbuds, rose of Sharon, lemonade berry, and sugarbush. So let's look at some of the scarification techniques in a little more detail. Mechanical abrasion involves scratching the hard seed coat deeply enough so that the seed coat is broken and water can enter the seed rapidly. But we need to take care not to shatter the seed completely by applying too much pressure or rupturing the seed coat so deeply that the embryo is damaged. We can use a sharp knife or a file to nick the seed coat of each seed individually, but this is obviously really tedious and time-consuming, so it's not particularly cost-efficient in a commercial nursery. A quicker method is to rub the seed between two sheets of sandpaper, and this is one of the techniques you'll be doing in the lab exercise. And there's a short video in Canvas showing you how to do this. If you have a lot of seed to scarify, you can use a modified rock tumbler where you've lined the drum with sandpaper, or you can put sand in with the seed. You can also purchase specialized seed scarification machinery, but it's expensive, usually in excess of $1,500, but worth it if you're a large grower needing to scarify large amounts of seed on a regular basis. After scarification, the seed can be sown in the usual way, or you can soak it in warm water for 12 to 24 hours to start the imbibition process, then sow the seed. Ideally, the seed will have been scarified well enough for it to take up water and germinate without the soaking process being necessary. Remember, every additional step you take to produce a plant adds to the cost of production, so don't do something if it's not necessary, or else make sure that the benefit of that extra work will outweigh the additional labor cost. On the right, you can see two sets of yellow bush lupin seeds. The lower set was scarified using a hot water soak for 24 hours, and the upper set of seeds was mechanically scarified with sandpaper and then soaked for 24 hours. You can see by the size of the two sets of seeds that the hot water treatment alone doesn't look as if it's been as effective as mechanical scarification. The seeds that were scarified before being soaked are swollen and are clearly well into the imbibition phase, and maybe even into the lag phase. If imbibition has even started in the lower set of seeds, it's certainly not visible yet. Many species found in chaparral and forest plant communities in Mediterranean-type climates especially, which includes most of California, are called fire followers. This refers to plants whose seed needs the heat of fire and, in some cases, exposure to smoke in order to germinate. Some plants need the heat from fire to release their seeds. For example, some conifers with serotinous cones, such as lodgepole pine, and some banksias and eucalypts also need the heat of fire. These plants have seeds that are sealed into their cones or fruit by a thick waxy resin that's melted by the heat of fire. In the nursery, we can replicate the heat of a fire by placing fruit in the oven at around 390 degrees Fahrenheit for about 10 minutes. Banksia follicles, which you can see on the right here, can be toasted over a fire like a huge s'more until they open to release the seed. But I definitely don't recommend trying this method in California during the dry summer and fall months because of the risk of wildfire. Acid scarification treatment mimics the action of acidic digestive juices on seed as it passes through the gut of birds or other animals. Concentrated sulfuric acid is usually used in a nursery or research situation, and the length of time that the seed is soaked in the acid varies with species from just a few minutes to several hours. And it's really important to rinse the seed thoroughly before sowing. It's really important to remember that using concentrated sulfuric acid is extremely hazardous. And here are a few guidelines for working with concentrated sulfuric acid. Never, ever, ever add water to concentrated sulfuric acid. It'll cause a sudden exothermic, which is a heat-releasing reaction, and will probably result in acid splashing up onto you, probably into your face or onto your chest. Always make sure the seed surface is completely dry when you add seed to acid. Always wear acid-resistant PPE, that's personal protective equipment, including acid-resistant face shield and goggles. Use acid-resistant containers and stirring rods. A safer option is to use less concentrated acids such as citric acid or acetic acid, which is vinegar. But you'll need to soak the seed for longer if you use either of these two acids. The seeds of some fire followers need to be exposed to the chemicals in smoke in order to germinate. For many years, it was thought that it was just the heat of the fire that broke seed dormancy in plants that are fire followers. But in 1990, two South African plant scientists, Drs. Boucher and Delange, discovered it was actually chemicals in the smoke that induce seed to germinate. Smoke can contain as many as 5,000 different compounds, and the science of the interactions between any of these and seed germination aren't fully understood. It's widely accepted, though, that smoke is beneficial for overcoming dormancy in many species, and also acts as a germination enhancer in species that aren't dormant, so species that are just quiescent. There are several ways in which we can apply smoke treatment to seed. One of the easiest and safest ways is to use seed primer smoke discs that are pictured here on the right. These are just discs of absorbent paper that have been impregnated with smoke. You place a disc or just a quarter of a disc in a container with about 10 milliliters of distilled water. Add the seeds and leave them to soak in the smoky water for about 24 hours. Then sow them in the usual way. You don't need to rinse them. We can also use liquid smoke to make a smoke water solution, but make sure that you buy one that's all natural and has no artificial ingredients. I think I bought the one pictured here on the right from one of the New Leaf stores. To use liquid smoke, you make a 10% solution of liquid smoke by mixing 1 part liquid smoke to 9 parts of distilled water. Place the seeds you want to treat in a container, cover them with the liquid smoke solution, and then leave them to soak for between 12 and 24 hours. Then sow the seeds in the usual way. You don't need to rinse them. Thirdly, you can make your own charate. This is a method I don't recommend trying during California's dry months, and of course you should be super careful at any time of year. To make charate, you collect some stems of plants from the same plant community as your seed is from. So if you're trying to germinate seeds from California chaparral plant communities, the most common plant to use for charate is chamise, which is Adenostoma fasciculatum. You cut the stems up into short sections, then char them thoroughly with a small blowtorch. Once the charred stems are cool, chop them up finely in an old food processor or a blender. Then sow your seeds and sprinkle the charate over the top of the plug tray or flat that you sowed your seeds in. And chemicals from the smoke will percolate through the substrate to the seed with the irrigation water. Make sure that you have the landowner's permission to collect any plant material you use for charate, and remember that it's illegal to collect any plant or animal material from public land. Examples of California native fire followers that need smoke treatment are many members of California's chaparral plant communities, including island bush poppy and the inland bush poppy, Matilaha poppy, and woolly blue curls. Examples of popular Australian plants needing smoke treatment are some of the grevillea and some of the banksia. And many plants from South Africa's fynbos plant community need smoke to germinate. And popular examples include the leucadendrons, the restios such as chondropetalum, elegia, and rhodocoma, leucospermums, and the proteas. But Remember that a lot of the leucadendrons, leucospermums, and proteas in cultivation are hybrids. And you're going to need to propagate these from cuttings if you want to retain the characteristics of the parent plant. Cold-moist stratification is a technique we use to mimic cold winter conditions in the wild. And depending on the native origin of the plant, the period of cold stratification may range from just a few days to many months. We can also sow seed and germinate it outside at the appropriate time of year if our local weather conditions are similar to those in the plant's native range. In a nursery, though, we're often wanting to sow seed outside of its normal cycle. So that's why techniques for breaking dormancy are so useful. We want the seed to germinate at our convenience, not when it's convenient for the plant. The photos on the right here show a couple of California native herbaceous perennials that grow natively in Santa Cruz County and beyond. Aquilegia formosa on the top right, the western columbine, and Douglas iris on the bottom, Iris douglasiana. Seed of both of these species would germinate in our area without any treatment if we sowed them and then left them outside over the winter. But germination may be sporadic and slow. We can obtain a much higher germination percentage and faster germination rate by cold stratifying the seed first. Western columbine for just 7 days and Douglas iris for 30 days. The photo on the right here shows bags of seeds of some California natives that are being cold stratified. And the bags contain our locally native winter deciduous shrub, ocean spray, and a herbaceous perennial with bright yellow daisy-like flowers, narrow mule's ears, that grows in grassland plant communities. So to cold stratify seeds, what we do is we place perlite or potting mix in a plastic baggie. Then we add the cleaned seed that we're wanting to grow to the perlite or the potting mix and mix it all thoroughly so that the seed is evenly distributed through the perlite or potting mix. Then moisten the whole mixture thoroughly. Don't saturate it. Tilt the bag to see if any excess water drains, and then pour off any of that excess water that's accumulating in the bottom of the bag. If you have water pooling in the bottom of the bag and leave the seed to stratify, you're probably going to find that the seed will rot. So pour off that excess water, and you may need to mix the seed and the perlite again just to make sure that everything's thoroughly mixed. The seed should have good contact with the perlite or the potting mix in the bag. Add a label to the bag with the name of the plant, the source if applicable, and the date that the seed is going into cold stratification. And then place the seed in the fridge. Never put it in the freezer. Create for yourself a calendar that shows the dates seed should be removed from cold stratification so that you remember when it's supposed to come out. And then check your seed regularly and remove and sow all of the seed in the usual way if any of it starts to emerge, even if the anticipated period of stratification hasn't finished. If you have enough space, then you can sow the seed first of all directly into a plug tray or other container, cover them, and put the whole tray in the fridge. You need to cover the tray to make sure the potting mix doesn't dry out in the fridge. And larger nurseries probably have a walk-in cooler for stratifying large quantities of seed. Finally, cleaning and leaching. Cleaning refers to the removal of fleshy parts of the fruit around the seed, and leaching involves thoroughly rinsing the seed in clean water to leach out any inhibitory chemicals in the seed coat or seed that may prevent germination. Again, these inhibitory chemicals are a survival mechanism in the wild and ensure that the seed will only start to germinate when sufficient rain has fallen and the soils are moist enough for seedlings to establish. There are two ways in which we can leach seed at home or in the nursery. One is to just soak the seed in a container of water, change the water every 12 hours or so, and repeat this process for several days. The second method is to wash the seed under constantly running clean water for 1 to 2 days. Clearly, this is incredibly wasteful of water and completely inappropriate in most parts of the world, not just California, where water's a valuable and scarce resource. One way around this is to put the seeds in a muslin or cheesecloth bag and hang the bag in the toilet tank. So note, this is hanging it in the tank of the toilet, not the bowl of the toilet. The seeds will be soaking in the water, and the water will be changed every time someone flushes the toilet. I read about this method some time ago being used somewhere in Australia, and it seems like a really practical solution. So a couple of examples of plants that benefit from leaching processes include the Australian plants Eriostemon, which is waxflower and pictured on the right here, and Australian fuchsia, Corea. And lastly now, I just want to say a few words about germination enhancers. In a commercial nursery, germination rate and uniformity can be as important as germination percentage. And there are several treatments we can use to promote germination. I'm not talking here about treatments that are necessarily used to break dormancy, but treatments that we can use on quiescent, so non-dormant seed, in order to achieve better and faster results. A fairly commonly used germination enhancer is the plant growth regulator gibberellic acid. This is available as a powder, and then you make a solution with distilled water at the appropriate strength, which is then used to soak the seeds prior to sowing them. Several penstemon species benefit from a soaking in gibberellic acid, and you can buy this product from J.L. Hudson Seedsman, who's based in La Honda, not too far away from us. I've already talked about smoke as a treatment for breaking dormancy. Well, we can also use it on quiescent seed to enhance germination. There's been a lot of research showing that it enhances germination in maize, rice, tomato, lettuce, some grasses, and some gymnosperms. And this is a treatment you may want to experiment with at home to see if it works for you. And thirdly, hydrogen peroxide. This is produced naturally by the seed during germination, and it's thought that it acts as a signaling function or a trigger. We can use a solution of hydrogen peroxide as a seed soak prior to sowing. And there's also some evidence that it kills surface-borne pathogens on the seed, so it reduces the risk of bacterial and fungal disease developing in the propagation environment. Now let's summarize what we've covered so far in this unit. Non-dormant seed is described as being in a state of quiescence and has the capacity to germinate readily when we give it the right environmental conditions of water, temperature, light, and oxygen. In contrast, a dormant seed can be defined as one that does not have the capacity to germinate in a specified period of time, under any combination of normal physical environmental factors that include temperature, light, dark, etc., that otherwise is favorable for its germination. Seed dormancy is one of plants' many survival adaptations to adverse environmental conditions. There's still a lot that we don't understand about the physiological processes involved in seed germination and dormancy, but we do know that they differ between genotypes, and we know that there are 5 classes of seed dormancy, and that seed can sometimes exhibit more than one class of dormancy. The techniques used in nurseries to break dormancy are mostly based on empirical evidence and include scarification, stratification, smoke, leaching, and application of growth regulators. And sometimes using more than one technique is needed. Some of the techniques used to break dormancy can also be used on quiescent seed to achieve a higher germination percentage, a faster germination rate, and a greater germination uniformity.